Hello, I'd like to introduce myself before we launch into the lecture. I'm Dr. Robert Sackstein. I have an MD and a PhD. My PhD is in immunology. I'm a professor at Harvard Medical School. I'm a clinician in the bone marrow transplant program of the Brigham and Women's Hospital and the Dana-Farber Cancer Institute. I run a basic research laboratory devoted to glycoscience-based inquiry of human disease, in particular a laboratory that's focused on translational glycobiology. I spend approximately 60% of my time in the laboratory, approximately 20% of my time taking care of patients, and 20% of my time in educational activities. I see it as my job, my purpose, my future, to train the generation of scientists and clinicians that will follow in our footsteps to provide the answers to make future generations feel less suffering. That's the purpose of the work. So with that, we'll begin this lecture. The topic I'll be presenting is the evolution of 20 years worth of work having to do with modification of cell surface glycans for an intended biologic effect. The title of the presentation is Glycosyltransferase Programmed Stereosubstitution, otherwise known as GPS. Directing cell migration via translational glycobiology. Before we begin, I'd like to make a disclosure related to intellectual property, which is required of my institution. In accordance with the United States National Institutes of Health policies and procedures, the Brigham and Women's Hospital has returned all rights of intellectual property related to the molecule H-cell and to the GPS technology to the inventor, which is me. Today's discussion will be presented in outline form with 5 broad topics. We will begin with an understanding first of the topic of translational research. We will provide an understanding of homing and the biology of cell migration. We'll describe the molecular effectors of homing to marrow. We'll then illustrate how vascular delivery of stem cells can be programmed via glycosyltransferase programmed stereosubstitution, otherwise known as GPS, and also describe the step 2 chemokine bypass pathway. And finally, we'll be introducing the field of translational glycobiology. So let us begin. Translational research, by definition, is a form of applied research as opposed to pure research, wherein the specific goal is to alleviate human suffering by providing basic science inquiry, thereby providing mechanistic insights applicable to the patient. Ideally, this is an inspired process. I want to emphasize the differences between pure research versus applied research. Pure research might be an understanding of why a butterfly's wings are colored a certain color. It seems, at least on the surface, that that would have very little to do with human biology. Maybe someday someone would find a dye that would be beneficial in providing a color to the hair or something like that. But with regards to translational research, we're looking at human suffering, a human disease, and attempting to identify the molecular basis of that disease with basic science studies in the laboratory, which will then translate back into the care of that patient. Like I say, it is ideally an inspired process. So here's a slide of me in the operating room performing a bone marrow harvest. I'm clinically a bone marrow transplant physician, and the topic we're going to be presenting in this discussion has to do with my role as a transplant physician. Here I'm performing a harvest, which is taking a large bore needle, 11 gauge needle, introducing it into the superior iliac crest in the lower back of a donor. I then extract the marrow material, and back here a colleague is setting up a filter set where we will pass that marrow material through a filter that will remove bony spicules and fat, which would otherwise cause an embolism in the recipient, and collect the cells through that filter at the bottom here into a reservoir, which will then be injected into the recipient. We have done bone marrow transplant for a variety of indications, including hematologic malignancies, and also this condition, which is osteogenesis imperfecta. This is a child who's suffering with type 3 osteogenesis imperfecta, one of the worst subtypes of the disease. This is caused by a mutation in the collagen gene, which causes disorganized formation of bone. And this poor child will be tortured for her entire life, which will be shortened by this disease. These patients, unfortunately, will only live maximally to about 20 years, many of them less than that, because the bones in the thorax cannot form properly, so they can't even breathe. This child will undergo at least 100 to 200 surgeries over the course of her lifetime to make corrections of these bony deformities, try to straighten out the bones with rods and so on and so forth. But it is simply a disease caused by bad collagen formation by the osteoblasts, the cells that make bone. Bone marrow transplants have been attempted in this disease. Unfortunately, they failed because of inadequate homing of the precursor of the osteoblast cell, called the mesenchymal stem cell, into the marrow. And that's going to be a topic we'll go into greater detail on today. As a general point, I want to emphasize, mice are not humans. It seems like something we would take as face value, but so many of our colleagues in science will do a mouse study and then claim that this is translatable to the human condition. It is not translatable. If you seek to study a human disease, especially a stem cell disease, then get human stem cells. Study the human biology. Investigate non-human stem cells only when there is no other choice. Well, in this case, we have a good choice because we can get the mesenchymal stem cell from the marrow and the fat, in fact, of a human. So let's talk about the adult stem cells that are present in the marrow. There's two principal subsets of cells, stem cells in the marrow. One is the hematopoietic stem cell, which is the cell that's best known to everyone. This is the cell that we seek to transplant when we do a bone marrow transplant. This cell can be identified by a marker called the CD34 antigen. And therefore, you can collect these cells, a sufficient number of those cells, to perform a bone marrow transplant. On the other hand, there's another adult stem cell in the marrow, the adult stem cell, the mesenchymal stem cell. This adult stem cell type does not have a marker where we can pull the cell out directly from the marrow, but it has one great advantage, and that is, though it is present at a very low percentage of the marrow cells, typically 0.01% of the nucleated cells of the marrow, it can be massively grown in culture and expanded in culture such that you can create a bank of these cells readily from as little as a teaspoon of marrow and as little as a gram of fat, because the mesenchymal stem cells are also located in fat tissue. Now, if you look at these cells that the mesenchymal stem cell can make, I want to highlight the fact that the mesenchymal stem cell is the precursor of the osteoblast, which is the cell that will make bone. So therefore, if we seek to cure osteogenesis imperfecta or other generalized bone diseases like osteoporosis, we need to harness the mesenchymal stem cell and get that mesenchymal stem cell to traffic back into the bone, i.e., to traffic back into the marrow, so that cell will then differentiate into a bone-forming cell, the osteoblast, and then create the proper bony matrix. So the fundamental question we have is, how can we direct systemic distribution of infused mesenchymal stem cells from the bloodstream to cure disease? Well, simply put, it's analogous to driving on a freeway, an autopista, an autostrada, an interstate highway. When you're traveling at 150 kilometers an hour, 100 miles an hour on one of these superhighways, when you want to exit that highway, you have to apply a brake, a braking action that'll let you get off that exit ramp. If you try to take that exit ramp at that speed, 100 miles an hour or more, 150 kilometers, let's say, per hour, you will crash. Well, a cell in blood flow can move as fast as 6 times its diameter in a microsecond. So if that cell seeks to get out of that blood flow and into a tissue, it has to slow down. It has to apply a molecular brake, which will allow that cell to firmly adhere to the area that it needs to go into and then transmigrate into that tissue, very much like a car has to do so in exiting an autostrada or an interstate freeway. So what we seek to do is to optimize these molecular exit ramps that direct the stem cell migration into a target tissue under hemodynamic, that is, blood flow conditions. So injections of stem cells have already been performed in myocardial infarcts. In those situations clinically, people have put the stem cells directly into the heart with a needle. I will argue that that is not the physiologic way of doing this, and I'll tell you why in the next slide. But the heart can tolerate these injections because the heart is basically a big muscle, so it's a firm tissue with strict anatomic boundaries, which can allow you to intersect or intercede directly into that tissue, injecting directly into that site. However, many tissues don't have these types of anatomic boundaries. Importantly, the lung, it's just a large air sac. You cannot inject specific areas of the lung, and certainly for a generalized disease of the lung, you couldn't inject the entire lung anyway. Other diseases like the skin, if you have third-degree burns over 90% of your skin, you couldn't inject every square micron of skin to get a stem cell to regenerate skin under a burn situation. And the colon is the same situation for patients with inflammatory bowel disease. It's too diffuse. Other diseases of the central nervous system, such as multiple sclerosis, too many lesions, too many chances of causing morbidity or trauma to the tissue, injecting those sites, it's impossible to do that. For many diseases, direct injection is not practical. So let's go into the major problems and limitations of the direct injection approach. Well, first, as I mentioned, it's only useful for tissues with strict anatomic boundaries. So you can use it for the heart, but you couldn't use it for the lung, for the central nervous system, for the GI tract. It's invasive and thus can be associated with procedure-related morbidity. Importantly, by introducing these cells under hydrostatic pressure, that is, in a needle, into a site, you actually could disrupt, under the pressure situations of that injection, the incipient repair process and or accentuate the inflammatory reaction at the site. Also, the direct injection of a stem cell into a hostile, that is, inflammatory site, would put that cell at a disadvantage in knowing what to do, number one, and number two, whether it could survive. Low pH, release of proteases and glycosidases and elastase, things of these natures, these enzymes that will disrupt the matrix of that tissue would prevent that cell from taking a foothold and possibly even surviving at the site. And certainly there's no role in multifocal or generalized diseases and conditions such as osteogenesis imperfecta, a generalized skeletal disease, or osteoporosis, another example of a generalized disease, or multiple sclerosis, or muscular dystrophy, or any of these diseases which are quite generalized. I want to point out, importantly, the difference between the localization of a cell and the homing of a cell. Localization is not homing. Localization is the process by which a cell is injected into the bloodstream and gets trapped in a particular blood vessel or endothelial bed. It's gotten there by retention, not by an active process where the cell actively sought or was directed to get to that tissue site. On the other hand, homing, which is truly migration of the cell, is an active process where the cell is recruited to the tissue of interest and it's lodged there. By physiologic mechanisms, the cell transmigrates the endothelium and moves partitions within the microenvironment of the tissue to where it needs to be. So shown here in cartoon fashion is the force of flow that causes entrapment, generally where a cell will basically find a terminal vessel that cannot pass further beyond that spot and gets caught within that tissue just because it's caught inside the vasculature. On the other hand, homing is an active process where a cell in blood flow will come down onto the endothelial barrier of the tissue of interest, will begin to tether and roll, applying a molecular brake, and subsequently will firmly adhere to the endothelial surface and transmigrate. And we're going to go into great detail now on the molecular basis of this process. This process is best summarized by what's called the multi-step paradigm. In the first step of this process, step 1, a cell in blood flow has adhesion molecules which will be specialized to allow that cell to come down onto the endothelial surface and begin applying a molecular brake, a decelerative action, onto the endothelium itself. Thereafter, the cell will begin to roll on the endothelium, and if it encounters, usually, chemokines in that milieu, which are small molecular weight molecules we'll touch on in just a few slides, there will be activation through chemokine receptors, which are G-protein coupled receptors, of integrins, another class of adhesion molecule, which will then firmly adhere through binding to their co-receptors on the endothelium. The cell will firmly adhere to the endothelium and then transmigrate. So step 1 is the tethering and rolling step. Step 2 is the integrin activation. Step 3 is the firm adherence, and step 4 is transmigration and then subsequent partitioning into the parenchyma on chemokine gradients usually. So at the first step, the molecules that mediate that process are the homing receptors. So by definition, what is a homing receptor? Historically, back 50 years ago when they were first being observed, which was in the first setting, homing of lymphocytes to lymph nodes, they were thought to be molecules that directed a tissue-specific pattern of homing or cell migration. But more broadly, as we now understand these molecules biophysically, they are described as cell surface molecules that are effectors of step 1 interactions. That is, they are the molecular brakes. So shown here now is a table of all known homing receptors. By definition, the homing receptor is expressed on the circulating cell. Its co-receptor or ligand is expressed on the endothelial cell. So on the left panel here are all the known homing receptors. On the right side of the panel are the adhesion molecule receptors or ligands for the homing receptors. I want to point out here importantly, a number of these classes of homing receptors fall into the selectin ligand receptor ligand interaction family. And I'll go into this in just a moment. But I want to point out here this molecule CD44 and another molecule that's called H-cell. H-cell is a specialized carbohydrate modified form of CD44, whereas CD44 will bind to hyaluronic acid and function as a homing receptor. H-cell, its glycosylated counterpart, which is specialized to bind E-selectin, will seek out E-selectin. This is very similar to this molecular pair here, the PSGO1-CLA axis, where PSGO1, a molecule expressed on leukocytes and on hematopoietic stem cells, binds P-selectin as well as L-selectin, but its glycosylated counterpart specialized to display specific carbohydrate sequences recognized by monoclonal antibody, HECA452, which I'll talk about in just a few slides, will bind E-selectin. So CLA is a glycosylated variant of PSGO1. Now, importantly, the selectins and their ligands are the most potent homing receptors. And other molecules, the integrins, can also function as homing receptors. One powerful homing receptor of the integrin class is this alpha-4 beta-7 molecule known as LPAM1. But VLA4 and LFA1 are rather anemic homing receptors, and importantly can only really engage as molecular brakes if they're activated, though these other molecules do not require, the selectins and their ligands do not require an activation sequence. So let's talk about the step 2 effectors. Most generally, it's the chemokines. Chemokines are small molecular weight molecules, generally less than 10,000 molecular weight. Over 50 are characterized, and they direct transendothelial migration by functioning in the step 2, the integrin activation portion of this. The cells in blood flow will have receptors for these chemokines that are present within the endothelial beds of the target tissue. So they are effectors of step 2 homing cascade. They activate the integrins via the chemokine receptor engagement, leading to firm adherence of the cell on the target tissue endothelium. And some of these chemokines are constitutively expressed in selected target tissues, so they regulate physiologic trafficking to that tissue, whereas others are upregulated during inflammation. And in every case, they function as chemoattractants of cells onto the endothelial barrier, and then subsequently when the cell transmigrates into the microenvironmental niche of that tissue. There are chemokine gradients that direct cells to get there.